Okay, hi everyone. Thank you for the introduction, vote. Imagine that we have an AI that is trained on all public proteomics data. Imagine how it would look like. Maybe you can go to a ChatGPT-like window and ask a question, and it comes back with a hypothesis that is data-driven and brings you back to the original data. Or it can immediately do a meta-analysis and combine a lot of public data sets together. Now, we don't have that yet. Depending on your opinion of AI, that's a dream or a nightmare. I'll leave that up to you. But it's not because we don't have the AI technologies to do so, and it's also not that we don't have the data. We could do it, but there's something missing in the middle, and that's what I want to talk about today. I want to talk about three things. First one, of data, the repositories. Why are they becoming a data graveyard? Then second, community efforts that are ongoing to make our AI data, proteomics data more AI ready. And I want to end with an example of what we can do if you have this intersection. If you can connect your data, your public data, to your AI with Marker, which is a public data AI-driven biomarker discovery tool. All right. So most of you, computational biologists, you know what proteomics changes. This is the umbrella term for all our repositories, and we have been depositing our data in there for well over a decade. And there's a variety of repositories, and all of them see the same pattern. There's an exponential increase in the amount of data that's being deposited, but also in the size of these data sets. Now, why are we doing this? Why are we depositing our data there? Well, the easy answer is because we have to of the journals, otherwise we can't publish. But there has always been a bigger picture behind it. And the bigger picture is that this data is extremely valuable. And maybe we are creating data now that we don't have the tools yet to get everything out of it. So storing the data in the repositories has always been to facilitate long-term reuse. Now, after more than 10 years, has this happened? Well, there have been some knowledge bases. You might know Expression Atlas or Proteomics DB. They are all repository-driven. There's also tools that are being developed like MS2PIP and DeepLC that can predict fragmentation pattern and retention times, but these are also very, very technical. So where is the new biology? What is happening? Why is the AI not there? Why are there no multi-omics studies? I mean, the transcriptomics people have repositories, we have repositories. Why are we not connecting these two? And one of the main issues, you probably all know it, right? Metadata is extremely missing. And metadata is the essential context to make sense out of your data. It goes beyond what organism are we looking at or what organism part are we studying. It goes, what were your instrument settings? What was your experimental design? What are you actually studying? And how can you represent this in a structured manner that can be read by machines? Now, what you see here is me spending way too much time on manually annotating and measuring how much missingness there is. And in orange, you can see what is missing in Pride's dataset descriptors and what is missing in the publications. The higher the bar, the worse it gets. And it's not a happy picture. But this problem is known to the community, luckily, for a lot longer. And in 2021, there was UBIC and HupoPSI, so that's European Bioinformatics Community and the Proteomics Standard Initiative. They came together and they were like, okay, how can we present metadata in a structured manner so that it can be provided in a way that can be read by machines? And this is SDRF. Just a show of hands, who has heard of SDRF? Oh my God, I'm so happy. That's the majority. I love computational conferences. So you know that it presents everything you would want to know about your experimental setup. But another show of hands, who have ever tried to make one? And who has succeeded? That's so depressing. Well, if you ever tried to make one, I don't blame you. I also gave up still quite a lot of times. You have to go through a Nature paper, a GitHub page, browse all these different ontologies to try to find terms that match your research, Often there is no ontology term for it. You have to make a literal flowchart of your experimental design and then find a way to put it all into Excel, and we all hate Excel. So it is extremely laborious and hard. So after a lot of frustration and against the advice of my promoters, I made a tool for it. I was, okay, let's try and combine all these things, these ontologies, these guidelines. Even if you have local metadata, just chuck it all into one thing, and it's called LessDRF, because I want to spend less time on SDRF. And it's a Streamlit application. We've heard about Streamlit applications before. They're easy. You don't need to install anything. You can click your way through it, and it's a little bit less scary to the wet lab scientist. Now, that was cool, but you've seen the exponential increase that we have in data sets. LessDRF is not going to solve the insane amount of data that is currently in our public repositories. So how are we going to get the metadata for these data sets, for our historical data sets? Well, I've mentioned UBIC before, I mentioned HupoPSI before. The next things I'll present are extremely community-driven, with the first one being an effort between UBIC and NSEMS. And what we are trying to do, and we're getting rather good at it, is make an AI-driven metadata extraction pipeline. And it has 4 key parts. So the first one was like, okay, we need to learn robots, LLMs, how to annotate data. We have to compare it to humans. So we spent some time on gathering a bunch of papers, annotating them with humans, annotating them with a GPT, and comparing them to. Why? Well, we eventually want to train a large language model that can extract this automatically, but large language models require a lot of training data. And I am not convincing enough to convince like 100 people to annotate 2,000 papers. I can easily ask a GPT to do that, but then we need to know that the GPT is doing a good job. So that's the second part. We want to train an SDRF bird model or something else than a bird model. Still figuring that one out. And then in parallel to that, we have a pipeline that extracts technical metadata directly from the MZML files. Because what I've seen during annotating is that people can write mistakes. Metadata is extremely scattered, and a lot of times people send their samples to the proteomics core, and then they get the data back, and then the data gets sent to the statistician, and then the original researcher has to like scramble everything together, and they might think that it was run on a timsTOF, but it was an Astral. Data, however, data never lies. So we wanted to add this technical pipeline to it to really be sure of what is going on. And then the ultimate goal will be to integrate this all into an updated version of LessDRF, in which you can just click, okay, this is my manuscript, these are my MZML files. Now get me as close to a complete SDRF as is possible based on what I have. So this is work that is done mainly by Harry, who's over there, and Katarina's over there for so detailed questions, and then Armin and Ian as well from Penn State. So the first part, I talked a bit about the text mining annotation thing, but this is in a bit more detail. So what we did is we gathered a corpus of TXT files that contains abstracts and material methods of papers. And that was annotated by 3 different groups of people, I would say. The first one was a group of 8 people from UBIC, and they all did a subset of these papers. So we could start, okay, what is the overlap between these 2? Then all these papers were also given to a GPT model. But we also wanted to know, okay, if we compare the GPT that has like this consistent annotation, we kind of also need a human that has this consistent annotation. So Ian, which we call the single human, Ian annotated all like 40 papers. He was actually stuck during a hurricane when he did so. He had no electricity, so he was like, I can as well annotate. So that's how deep you have to go before annotation. So we have these 3 sets of annotations, and we can start comparing them. Okay, how well do they agree with one another? And use that to eventually fine-tune a GPT model to get optimal agreement. And that is then used to train our corpus data for the next part. I'll come back to the fuzzy matching part. Now, indeed, how does that look for some of you who might have never done manual annotation? This is an abstract, and you are sitting in front of your computer, and every time you see human, you click and you say, this is an organism. Then you see heart, and you click and say, this is an organism part. It's a bit like key value pairs in dictionaries, but here we call it entities that get a specific label. We have around 60 different labels in SERF that we train on, which is quite a lot. And we write things in a very, very different manner, and we annotate things in a very different manner. So what we immediately started seeing is that what annotator 1 says is the organism is mouse, the other one will say it's murine. And the first one will say, yeah, the organism part was prefrontal cortex, and the other one will say, no, it's brain. So if we would make semantic comparisons between these annotators, all scores would say, yeah, no, they don't agree with one another. So in parallel, in addition to the semantic comparisons that are standard in text mining, we also did embeddings of these terms, because then you can see that mouse and urine will have a close embedding to one another. So we also took the average embedding distance into account when doing these comparisons. Oh, wait, I have a pointer now. Okay. What we can see here is a UMAP clustering of our annotated documents. On the left, they are colored according to annotator, and on the right, colored according to document. Why did we do this? Well, I was quite fearful that we would see one cluster that says GPT annotated. But you can see that the green dots, which is the GPT, are scattered amongst the 3 clusters. What you also can see is that the documents don't cluster together. So it's still in the process of figuring out exactly why there are 3 clusters. But what we can currently see, it has to do with certain experimental setups and the way that annotators annotate, technically. But how well do they agree with one another? Well, if we look at experiment level, document level agreements, we have our precision, our F1, and our Jacquard score. And in green, it's the fuzzy metric, and then in blue, it's when we look at embeddings. And then you can see, okay, semantically, we might not always agree, but conceptually, we are. We can get this on a specific inter-annotator agreement. So we can see how much Harry and I, for example, agreed in annotating, and we can start shaming people when they do a bad job, which we didn't, but we could. But what I want to focus on is what you can see at the top here. This is the GPT. So the first few rows are the GPT annotations. And again, they are not drastically different from the humans one. So we're okay, it's a bit underperforming compared to humans, which we expected, but it's not doing anything weird, it's not doing anything bonkers. This is on a document level. Some things are easier to annotate than others. It's easy to say that they studied human. It is quite hard to say how many technical replicates they had. So we did the same analysis again on specific entity levels. This is what you can see. So performance drops and gets spread out a lot more. Why is this happening? Well, a few reasons. For example, there are a lot that I colored in red, and these are technical things like fractions, technical replicates, biological replicates. They are very, very hard to grasp how many technical replicates they had belonging to each condition. We've tried it, and even as humans, we could not always agree with one another. The one in pink, for example, things like tumor site and treatment, we just did not have a lot of training points for. So this just shows that there is a lot of variability on the entity level. Okay. Then we wanted to know also which GPT is performing the best. So what we actually did is we compared Ian to the multi-human, so a single human to a multi-human, because we were like, okay, this is the upper limit of agreement we can possibly get. And then we wanted to see how far is a GPT off from this upper limit. And then you can see that at the top, the O4 mini model actually performed the best. So this is what we've been using for fine-tuning and that we will use to build the corpus. Now, an important thing to know about GPTs is that they make an embedding of your text and they can extract some data from it, but they can't pinpoint where exactly in the text they got that information from. So you can ask it, give me the coordinates where you found that the organism was human, and it will just make up random numbers. And that's quite annoying, because if you want to train a bird model later on, you need to give it a text, and then you need to say, organism was human and was found at from this location to this location. It really needs the coordinates, because a bird model, in contrast to a GPT model, looks at everything before and after it. So that's a bidirectional model, while a GPT just reads like a human from left to right, and that's it. So the bird model really needs coordinates. So what Harry has been working on the last few weeks is making sure that we have a fuzzy matching algorithm that can say, okay, we have these annotated texts. Now let's go back and see if we can find the coordinates back. And this is how we create our corpus for our SDF bird model. Currently, there are more than 1,000 papers that are being used to train the bird model. Harry promised me results, but I don't have them yet, so I will come back to you on that later. But the goal is eventually to not only look at manuscripts, but also look at, for example, supplementary files. There are LLM things out there like Dockling that can really easily take a PDF and transport it to a TXT file, which is readable by a bird model, and the same for PRIDE's descriptors, dataset descriptors. So the idea is to not only stick to the manuscripts, because a lot of the metadata will be in the supplementary files, sadly. All right, in parallel to that, technical metadata that can go as broad as you want. But we started focusing on two pipelines. One is Run Assessor. This is a pipeline that's originally developed by Erik Deutsch from the Peptide Atlas. And then second to that is Ian, who is working on a pipeline to identify organisms. So this is a list of all the things that Run Assessor can extract purely from MZML files. And it's quite cool that you can go beyond instrument information and acquisition mode, but it also starts to see, for example, based on reporter ions, was there any labeling used? Because sometimes they don't even mention if they used any labeling within the text. So you get multiple sources of metadata, and the ultimate goal is then to use that to build a probability metric of, we are this % sure that they use TMT labels. And then organism. You might think it's rather easy to read from a text what organism they used, unless they did an experiment in which they compared human versus mouse, and they don't say which raw file belongs to which species. So in that case, we have two different pipelines that Ian is working on. The first one is CompMS2. So that is a tool originally developed by Magnus Palmblad and Ben Neely, and that looks at spectrum similarity for species identification. So what we'll do is we'll look at, do we have any annotations, or do we have any model organisms that are expected to be in here? And then do spectrum similarity scoring to see how close they are to, like the experimental spectra are to the reference proteome of said species. If we are looking at a mixture of metaproteomics, crazy bunker stuff, we have no idea what to do with, we're gonna look into using Casanova together with Unipept to go more to the identification. So really go to peptides and then go to Unipept and say, okay, this peptide, combination of peptides, to which species does this belong? But this is only metadata. And metadata is not the only thing that is missing in the community. So within the Proteomics Standard Initiative, we started an AI working group, which I'm chairing together with Samuel Vine and Ralph Gabriels. And we're gonna focus on a lot more than only metadata. So the first thing we really want to focus on is, we are, all of us, developing AI methods. How are we reporting these? I've heard questions today about, but how did you do the tokenization? How did you do the embeddings? These things will be key to start reporting in a structured and consequent manner to know exactly what is going on. The second is, if everyone starts using AI on public data from these repositories, how are we going to assure an ethical and a privacy-aware reuse of data? There are a lot of biases in our public data that maybe not everyone is aware of, but most of our proteomics data comes from white males. So if you build a model on that is completely unaware of any gender, of any race, well, you're going to build in a bias, but how can we make these models aware of gender and race without compromising the privacy of the individuals? So that will also be one of the task forces, look into federated learnings for AI agents, etc. And then on the data level itself, we have 3 clear levels, with the first one being, of course, the metadata. This is where the UBIC will come in and the NSEMS effort will come in. The second being the data formats. Our repositories are filled with vendor formats. What if they stop supporting the conversion tools? What if at one point we cannot open .d files from Brooker anymore? What are we going to do with that? How are we going to prevent bit rot from happening? We're just storing them. The repositories are becoming graveyards. We need to find a way to sort this out before we lose data. And the last thing is we want to look at reprocessing tools. This should be something that everyone in the field can benefit from, and not only the computational scientist. So making tools that are easily accessible, that can do large-scale analysis without putting a heavy burden on the user of having a steep learning curve will also be one of our focuses. Now, we don't want to reinvent the wheel. A lot of these things have already been developed. OpenMS is doing things like that. We have Proteomics ML who's educating people. We have UBIC and NSEMS are working on metadata. So this working group really wants to be an umbrella to bring all of these efforts together. So if you would be interested in joining it, there's a link here. We're going to have our kickoff event somewhere in August. And then we're going to start really getting into the nitty-gritty of all of this. But why? Why would we do this? Well, I can give you an example from my research. There are a lot more out there, but I know this one the best. And that is because I spent, during my PhD, 6 months in COVID, in lockdown, manually annotating papers until I annotated 183 projects. And these projects were selected because they contained healthy human baseline tissue proteomes. And I collected a bunch of them. This was all reprocessed, I filtered them, I quantified it, and stored this all into a huge protein expression atlas. And this is more or less what it looks like in a t-SNE. Every dot is a raw file, every color is a tissue, and similar colors mean similar tissues. And what you can see is that it clusters nicely together, which makes sense. Your brain does not look like your liver. But if things cluster together, a lot of the times they classify also very well. So I trained a very simple random forest model on this. You can see the performance here. So on the y-axis, we have the predicted tissue. On the x-axis, oh, it's the other way. On the y-axis, you have the true tissue. And then on the x-axis, the predicted tissue. And where the diagonal meets, that means the model is doing a good job. The darker the color, the better the job. Again, it makes sense that that model can pull these apart. If you see syntaxin, you know that you're looking at your brain. Now, 2 things to keep into mind of this model before I go into all the fancy stuff I did with it, is that it's a random forest classifier, meaning it has a fixed feature space. It can only use the proteins it was trained on. If I present it with a new sample that has a very fancy isoform it has never seen before, cannot use that information. The second thing is, though, a random forest classifier is very interpretable. So you can get protein level interpretability. And this becomes really, really cool when you start giving it things it's not meant to know. I presented it with a small intestine sample from a patient with celiac disease. It has never seen celiac disease. So we got, and you can see here the tissues it predicted, and it was rather sure small intestine. Then it got confused. So it was nasal polyps, and then it went back to duodenum and colon and gastrointestinal related tissues. But so what is happening with these nasal polyps? Well, random forest, protein level interpretability. I could get these SHAP force plots out. And these force plots really show you in pink the proteins that say this is really pushing against the blue proteins that say this is not small intestine. And you can get that for every tissue. So here I have it for small intestine and nasal polyp. And you can see, for example, there are 2 proteins present that are very big pushing for this nasal polyp prediction. While small intestine, it's more a combination of smaller proteins. So I went deeper into this nasal polyp prediction. I wanted to see what was going on. And the cool thing is that this original study did a differential expression analysis. So they had healthy versus non-healthy tissue. And I looked at these nasal polyps protein and compared it to the original results. So the pink ones, the ones that say this is really, really nasal polyps, they were all immune-related. They were all immune-related, eosinophile-related proteins. And your nasal polyps are inflamed like 95% of the time. So it kind of makes sense that these ones got picked up. But the cool thing is, galectin-10 is a known celiac disease marker. And the tool here, the random forest, he picked it up. But the original researchers could not find it because it just fell below their arbitrary threshold of upregulation. Same for the ones that in blue. These are proteins that are very small intestine and gastrointestinal specific. And in the original research, one of them was downregulated, while the other one was again below this threshold of downregulation. So I started realizing, okay, I did more than building a classifier. This tool gives you not only a healthy counterpart to your disease sample, but it gives you a counterpart against every healthy tissue it has ever seen. So it gives you a healthy background of tissues and can find lower levels of patterns that you might not pick up on. So what do you do if you're a bioinformatician and you have something and you can like, oh, I can make a tool out of this? You give it a cool logo and you give it a cool name. So I called it ML-Marker. ML for machine learning, but also for molecular level interpretability. Why an octopus? Well, his name is Mark and he's cute and that's how far it goes. But I started seeing how far I can push this thing. Small intestine samples from celiac disease patients are still pretty similar to the small intestine samples it was trained upon. So what do you do as a bioinformatician when you make a tool? You try to break it. I presented it with cerebral melanoma metastasis, skin cancer that eventually got into the brain. ML-Marker has never seen cancer. It also has never seen the skin. I could not find sufficient healthy samples of skin to make a solid training on. So in this original research, they had two populations of these cancers. They had a population that responded well to the treatment, and then they had a population that responded very poor to the treatment. And when Zilla et al. They did their original analysis, they got this UMAP that showed two specific clusters, but it did not correlate to the response state at all. And they tried to do differential expression analysis, and they could pick up on 12 proteins, and they were incredibly frustrated because they got stuck. They knew something is happening in these patients. It has to do with an epithelial to mesenchymal transformation, so with the way that these tumors interact with the extracellular environment. But based on solely their phenotypic response, they could not see what was going on. I was okay, let's see what ML-Marker does with it. ML-Marker found the same two clusters. But if you look at the way these clusters are colored, they are colored based on brain similarity. And you could see a significant correlation between poor responders having a very high brain similarity. So the tumors that did not respond anyway to these treatments were just mimicking the brain a lot better than the tumors that did respond to the treatment. And if you use these two clusters to do the differential expression analysis, you get over 200 different proteins. So this is rather cool, like this is generating new hypotheses. But what is exactly going on? Are we only finding then brain-related proteins in these differential abundance proteins? Well, I told you that ML-Marker has a fixed feature space. The original samples had around 9,000 proteins, if I recall correctly, but ML-Marker only uses 5,000 of those, meaning that there were a lot of proteins in these samples that were not used to make this, but that were in the differential abundance list. So I did what any good wet lab scientist does. I made a gene ontology enrichment. And I wanted to see, are we only picking up on brain patterns or not? And you can see here that I have some, oh, well, the quality is bad. Okay, I have to take my word on this one. The red boxes, they are the brain-related processes. You can see it's not all red. I was suspecting it would all be red, but that is not the case. And what is cool here, you have some pink boxes as well. These are all processes related to the extracellular matrix. The exact hypothesis that your original researchers had and could not find in the data pops up here by clustering your data in another way, looking at it in another way, and using more orthogonal information that they previously had. Now, if you're a statistician, you might think, you did your clustering, and then you use clusters to do differential expression analysis. Does that count as double dipping, though? Is this a circular analysis of using the same data twice? Well, MLMarker does not use the protein abundances in itself. It compares it against its healthy tissue training data. Still, I was like, okay, fair point. Let's see. So what you can see here is you have the p-value distribution of, on the left, the proteins that are not within the MLMarker feature space, and on the right, the ones that are within the MLMarker feature space, with then in orange, the ones that have a significant log-fold change. There is a slight enrichment of differentially expressed proteins within the MLMarker feature space. However, if we then start looking at do these p-values and also correlate with the importance of these proteins in the MLMarker, that's what you can see here. So on the x-axis, you have the same p-values, and then on the y-axis, the feature importances within MLMarker. And there's no correlation. So this does not count as double dipping. MLMarker really presents you with a safe way of integrating orthogonal information and looking at your data from a new angle. But I still tried to break it some more. So I presented it with cerebrospinal fluid. It's a biofluid. It has never seen any fluids. I didn't train it on blood. I didn't train it on urine. I didn't train it on CSF. But it could still pick up pituitary gland proteins. It could still pick up brain proteins. And these proteins were known secretory proteins. They were known tissue leakage proteins, which I think is rather cool. Another one, single cells. There was a research from the Slavov lab in which they got testis samples from patients, went into the single cells of it, and then ran them on the mass spec. And I gave it to MLMarker, and even here, MLMarker was pretty sure that these were testes. There are some that the tool was not necessarily sure about, but that directly correlated to the amount of PSMs that were within those samples. So it's not magic. If you have a low quality sample, it cannot create something that is not there. But if you have sufficient PSMs within your single cell, even then it can start detecting tissue-specific protein expression patterns. So what do you do with that? Well, I made a Python package and I made a Streamlit application for it because I want web app researchers to be able to use it. And for example, this is what you can get when you go to the Streamlit application. It generates also figures for you. So on the top, you can see for every predicted tissue in green, the amount of proteins. I think the first one is pituitary gland. So you have a bar for all the proteins positively contributing to the pituitary gland. And then in red, the ones that say, aha, this is not pituitary gland. And you have this for every possible tissue. And then you can go into more detail and really start looking into, for example, on the left, you have pituitary gland and then brain, which proteins are responsible for this, and even get visualizations and scatter plots so you can compare multiple tissues against one another. The gene ontology enrichments are also there if you really, really want them. But I think this is pretty cool. You also have Mark floating around a lot, Clippy-wise, guiding you through your analysis. But this is, in my opinion, a good use case to show you how metadata is a bit like the Philosopher's Stone. So I'm a Potterhead. But just like the Philosopher's Stone touches things and turns it into gold, if you have metadata with your data set, it can literally turn into gold, and it can be reused in other approaches in a way that it can absolutely live forever, which is a bit cliche, but I think it gets the message across. So there's a list of names. As I said, most of this has been in a tremendous community effort, so I'm incredibly grateful to all the people who helped to this. And yeah, with that, if there are any questions, I'll be happy to answer them.
Great, thank you, Tine. We can kick off the questions with one from online. When you're reanalyzing and curating data, which ones are relevant? Will you also still include like the really old, low-res DDA data in those reanalyses?
Well, it depends what the purpose of the reanalysis is. So for tissue-specific patterns, I would absolutely do so, because your tissues do not change. You will just get a confirmation of patterns that you see also in the DIA. So if you see syntaxin in your brain, which is very high abundant, you will see it in an old and a new data set. If you are interested into more of the nitty-gritty and the very, very low abundant proteins, then I don't think they will add a lot. So it heavily depends on the application that you have. But I think it's always worth exploring also old data if you use new tools for it. Because maybe using a modern search engine of looking at modifications might get you some new information that was not seen by the original authors.
More questions?
Hi, thank you for your talk. I thought your comment on like the vendor formats was quite interesting, and I was wondering what your solution would be for if the vendors decide not to help.
Well, so it's a tricky one. What the PSI has been trying to do now is we developed MZ-Peak, and they have been actively talking to a lot of vendors which are more keen on supporting it, because MZML is very heavy. And if you go to Astral and timsTOF data, it just gets too big to store. It's not efficient anymore. MZ-Peak, there's a lot more interest for it. And then it would kind of be to the repositories to or enforce MZ-Peak submission or present the conversion to do the conversion within the repositories. Because it's the only way that we will be sure that we won't lose our data. So, yeah.
Hey, thank you. So I have a lot of experience with going back through transcriptomic repositories and kind of pulling analysis out of there. And I guess the quality of the metadata is, it's so person dependent. It's not even like data type dependent, right? Can you talk a little bit about what your experience was like in Pride, for example? I have a bunch of ideas of stuff I want to go and check in there, and one day I'll have time and I'll actually go and do it.
Wait, I didn't hear the
I'm curious of what's your experience with the Pride database and actually going through that and pulling studies out. Is it similar? Is it it's difficult, hard?
It is difficult. So there are multiple levels to Pride. Submitting your data to Pride is difficult because the submission tool was built before I was born in the early 90s. So that's already hard. So it doesn't really incentivize adding more labor to it, adding an SDRF to it, making it more hard. Then if you are beyond the submission process and you really want to say, okay, I just need to go to Pride and I need to find a healthy liver data set, if you don't know how to browse or use the API, it's very, very difficult. So what I've started doing now is just, I have a Pride Navigator tool that I just use in-house that scrapes all the SDRFs, and then you can browse through the SDRFs. But one of the things that we want to focus on with HUPO PSI, with the AI group, is get all this metadata in a centralized repository or like a storage place where it's easily searchable and indexable, because currently it's not really being indexed. So even if there is metadata, well, have good luck finding it.
Yeah, and I guess like in the data sets that end up in other places, because I know for one of our papers, we put it on Pride because a reviewer asked for it, but usually we'll just make everything available, through GitHub or some other kind of data repo. Do you have any, I guess, what's the coverage of proteomics data sets in those repositories? Do you have any feeling for that? Is there a bunch of data sets that are just other places you can't even see?
It's extremely scattered. So I've seen people adding the metadata to the supplementary files, which we try to tackle with the metadata extraction pipeline. We're not only going to look at Pride, there's also a lot of valuable data sets within Massive. But the issue with Massive is that the metadata format there is far from complete. Then if people start putting their metadata on GitHub, I have not seen this quite a lot, because it's mandatory to submit your data through the Proteomics Exchange portal in Proteomics. So it kind of goes there or in the manuscript. If people start putting it on GitHub, I'm probably going to lose it because I don't know how you will then eventually get to it. So yeah, well, have to keep an eye out if that ever happens. I might call you to know how you're doing it.
And I guess one last question. That's what would you suggest for if I'm submitting a paper next week? What should I be doing with metadata?
Submitting a paper next week. Well, that's the thing. People only start thinking about their metadata and the data when they are submitting the paper. And that is what annoys me, because your metadata will be scattered amongst the core facility, the statistician, you who did the experiments. It will be in your lab notebook. It will be in some emails to the core facility. It will be absolutely scattered everywhere. So preferably, I would want you to think about metadata before you start the experiment, similar to how you contact your statistician before, or should contact your statistician or your bioinformatician before you start doing your experiment, so you don't go to your bioinformatician and say, I have 3 samples, now do the machine learning magic. The same way you should start thinking about metadata then and like documenting it. If you haven't done that, well, good luck scavenging for it. LesDRF helps you quite a lot, and even incomplete metadata is better than no metadata or false metadata. So I would advise still try to fill it in as much as possible. And once we have the UBIC and SEMS pipeline up and running, it will be a lot easier to just chuck in whatever you have. But we're currently not at that stage yet. I hope by the end of the year, we'll see how that goes. But that would make it a lot easier. But some metadata is still better than nothing or false metadata. So, yeah.
And if I can briefly chime into the discussion, I think at this point in time, we're mainly asking the data submitters to do more work, right, to provide the metadata, which is not a winning proposition. But ideally, we ask this to be able to build tools such as ML marker that provides value to the community, right, to the users. So hopefully there will then become like a virtuous cycle of data and metadata being available, and then users driving value from it for their own work as well.
What I've also seen is that if you have a properly annotated data set, the amount of downloads you have to your data set increases dramatically. The amount of citations increases dramatically, and your impact factor as well. So what the repositories and the journals are looking at right now is, can we add a digital object identifier to data sets so we can start tracking the impact the data set has, similar to the way that a specific, like a very high valuable manuscript or article has impact. So that is something that is moving with the journals. What is moving with repositories is controlled access. Clinical proteomics is rising, and it's going to keep on rising. And preserving privacy in the GDPR world is going to be more important than ever before. So Pride is going to move to a section that has controlled access, meaning you will not be able to access it without approval from ethical committees, original authors. I'm not sure about the details. What I do know is that every data set within the controlled access repository will need a valid SDRF, otherwise you cannot submit it there, because you cannot assure that you have the privacy controlled if you don't have the SDRFs to keep track of it. So repositories and journals are both moving towards realizing the value of metadata, and even if users don't realize it by AI tools being developed that add value to their own research, at one point it will be inevitable. So it's better to get ahead of the curve, because we will all have to annotate at one point or another.
All right, we'll do another question over there.
Yes, thanks for a great talk and advocating for all this data management and submission. Yeah, definitely a big chunk of work for everyone, and I totally agree. Like, if we just push the burden to the original data submitter, it won't be very sustainable. And yeah, I see a lot in my organization as well. So just a thought, like, is there some possibility, or do you see some of the caveats, like if we include the metadata into the original data format, and then maybe create some new data standards? So instead of separating, for example, NGML from the SDRF into two separate parts. If they are together, then maybe there's a more tight connection between them.
So if I get it correctly, what you mean is that there will be different sources of metadata coming in if we do retroactive annotation?
So right now, I feel it's like we have the data format already there, and then we try to supplement new information that we think is useful by SDRF. But is it possible that we can combine them and then create a new standard? So then it's similar to these days when people need to submit, there's the MZ tab, but actually MZ tab already have some metadata.
Yeah, so we don't want to create a new format. There are plenty of formats, and there are plenty of formats that are not actively being used. So creating a new format is just completely out of the picture. We're gonna stay with SDRF, try to make it as efficient as possible. MZTab is more extensive and contains more detailed information. But in the proteomics community, it's not very adopted as far as I can see. I know metabolomics is a lot more attentive to that. Now, what I do think, it's the JPost repository that started a journal in which if you annotate a data set, you can actively publish that SDRF annotation as like a little technical paper. And the original researchers, they get like a ping to say, hey, someone annotated your data set. Could you maybe check if what is happening is correct? And I do think that is a good way of making sure that even if people are retroactively annotating, that the original researcher knows what is happening, can check it. If the PhD student left the lab, so be it, then we might be stuck with incomplete metadata annotation forever. That, well, that's often when I email office a lot, and they're like, can I have the SDRF for that? They're like, the PhD student left. But what JPost is doing is probably a good way to incentivize people to annotate and get the original authors involved. But we're not going to create a new SDRF format or flavor to it. We just try to see how far we can get it and how far we can get it adopted as it is.
All right. Thanks a lot, Tine.
Thank you very much.